This is the beginning of the streaming session. We'll mute the mic here in a minute. We'll open it back up once we start the session.
All right, folks. Thanks for making it out in the cold, and I'm here to help. What questions have you got to get ready for this test on tomorrow morning? Yeah, bright and early, 8 a.m. You know, the good news is the weather forecasts have taken a turn for the better. I don't think we have to worry too much about storms and such. But how do you like the room? Pretty cushy, huh? I was saying this is the very first class I ever taught at Wright State was in this room. And I'll tell you, back then, that was a long time ago, like 93, 94, something like that. But back then, I think there were more seats. But, you know, I know the walls haven't changed. I think it might be that I was just more intimidated by large numbers of students at that time, because I remember walking in and taking that sort of secret access thing down to the front of the room, looking out from behind the curtain like, oh my gosh, that's a lot of people. Now, older and wiser doesn't mean that much to me anymore. But anyway, so I'm trying to break the ice here. You guys got questions for me? Shout them out. Raise a hand. How can I help you? I think question, yes.
The definition of these types of RNA, the ones that are going in.
Sure. So the question is, what's up with all the different kinds of RNA? My paraphrasing of it. So, on some of the old exams, you'll see I give you 5 choices of different kinds of RNA, and I made it a point to at least mention each of those 5 times during the course of the lectures, so that question could be relevant. In no particular order, I'll try and rattle them off, and if I miss one, let me know. So, messenger RNA, mRNA, M for messenger. That's what it is that ribosomes translate. And so in the case of prokaryotes, that is the same thing as the primary transcript. Again, meaning that as the RNA polymerase is transcribing, ribosomes are right behind it, translating that messenger RNA into a protein. In the case of eukaryotes, the messenger RNA is still what it is that the ribosomes are translating, but it's the processed version of the RNA that gets out into the cytoplasm. Because in eukaryotes, transcription and translation are separate, and translation's only happening in the cytoplasm. When an RNA molecule that ribosomes are going to pay attention to is out in the cytoplasm, that's when we call it a messenger RNA, because at that point we know that all the processing is complete. And so that begs the question then, what do you call the RNA inside of a eukaryote before the processing is complete? And that is an HN RNA, or a heterogeneous RNA. You could also call it the primary transcript of the RNA polymerase, but the HN RNA may not even have been completely transcribed yet. The RNA polymerase may still be chugging along, or it may have released it, but it's going to include some things that won't make its way out into the cytoplasm, or won't make their way out into the cytoplasm. So things like unspliced introns will still be present, and things like the nucleotide sequences beyond the polyadenylation signal might still be present, meaning that splicing and polyadenylation may not have occurred yet. So it's the unprocessed primary transcript in a eukaryote. When the processing is complete and that RNA gets exported into the cytoplasm, we don't call it an HN RNA anymore. Now it's a messenger RNA, or an mRNA. Then the other three, and there are more, actually, but among the other three are things like ribosomal RNA. So ribosomes are made of a large subunit and a small subunit in both prokaryotes and eukaryotes. In both prokaryotes and eukaryotes, inside of both of those large and small subunits are RNA molecules. And it's not a trivial amount of RNA. In the old days, like when I was taking freshman biology, that ribosomal RNA was described as a scaffolding around which proteins were assembled. Well, you know, as people are looking more carefully at this, they're seeing that the ribosomal RNA actually accounts for more mass of a ribosome than those proteins do. And so there are a lot of implications that there's a functional significance beyond just providing a place that those ribosomal proteins can sit on. But so that's a ribosomal RNA, 2 or 3 different kinds, 2 in the large subunit, 1 in the small subunit for both prokaryotes and eukaryotes. You don't need to know those kind of details, just the fact that, oh, this is the RNA that's intrinsic to the structure of ribosomes. And then tRNA. I spent a good 15 minutes talking with you guys about tRNAs, as I recall. They've got that sort of classic image of a cruciform structure that no one in the class confessed to ever having seen before. I showed it to you. So I don't know if the word classic really applies, but nonetheless, they have an iconic image, at least to those who've been exposed to molecular biology. But the deal with a tRNA is those are the things that bring amino acids to the ribosomes, and they have an anticodon loop that base pairs specifically with the triplet codons as a messenger RNA is being translated. So each tRNA will have a specific anticodon. Depending upon which tRNA and which anticodon it has, a particular amino acid will be attached at its other end. So those are tRNAs. And then I think the 5th on the list is probably snRNA. Those are small nuclear RNAs. Those play important roles in splicing. They're intrinsic components to spliceosomes. I think that's probably enough detail about snRNAs for the purposes of this class. There are many other kinds of RNA that molecular biologists talk about. SNO RNAs, SNO, small nucleolar, so their location is limited to the nucleolus. There are silencing RNAs, siRNAs, antisense RNAs, so there's a whole bunch of others, and they each get their own little 2-letter or 1-letter lead up. But those are the 5 big ones and that are at least relatively important for a class like this. I'm still fighting that laryngitis I got just before Thanksgiving. How about that? Yes?
There was a question on one of the tests, and it's talking about, it says, which of the following is a correct description of the 3' end of the DNA strand? And it was the 3' hydroxyl group is not attached to anything but a small sugar. And I was a little confused because I thought that, like, that's where it's not attached.
The 3' end. You know, I have a vague recollection of somebody showing me a question like the one you're reading and saying, oh yeah, that was wrong. But I don't think I don't think you're reading to me from the one that's wrong. So I tell you what, let me just work through it and we can maybe figure it out together. So ribose is a 5-carbon sugar. You can start with an oxygen up there as I draw it out. Wasn't pretty, but it'll work. And there's a carbon up there. So this is the 1' carbon, the 2' carbon, the 3' carbon, 4', and the 5' is up there. When you put phosphate groups on a ribose sugar, and so the nitrogenous base gets attached to the 1' carbon. The 2' carbon is what lets you distinguish between RNA and DNA. DNA is deoxyribo, RNA is ribose, and so it'll have a hydroxyl group, whereas DNA will not. It'll just have 2 hydrogens off of that 2' carbon. The 3' carbon is going to have an OH associated with it, and that's specifically mentioned in the question that you're asking about. And if you were to add another nucleotide onto this to make it a dinucleotide, so there's one nucleotide I've sort of sketched out for you here. If we made it a dinucleotide, that next nucleotide would come in over here, and you'd make a phosphodiester bond between this 3' carbon and the 5' carbon on the incoming nucleotide. And so the phosphate groups would all be attached here. If it was a nucleotide triphosphate, you'd have the 3 phosphates there. So, does this help then with your question? What was the question exactly? There's nothing attached to the 3' hydroxyl?
It says it's not attached to anything but its own sugar.
The 3' hydroxyl is not attached to anything but its 3' carbon, I hope, as opposed to sugar, right? But so if we're focusing our attention on that hydroxyl group, it's not attached to anything but the 3' carbon. So this is the 3' end of a DNA strand or an RNA strand. That's the 3' end. The 5' end is going to have a phosphate group or 2 or 3 attached to it. Yeah. Sure. I love talking about the lac operon. You sure you want me to talk about the lac operon? This could go for a while. All right. I'll leave that there. It might come up again at some point. But so for the lactose operon, here's the lay of the land. All right. You've got a promoter. You've got an operator. You've got a set of 3 structural genes, Z, Y, and A. And I didn't tell you guys anything about this, but you came, so this is free knowledge for you. At the end of the whole thing, there's a lowercase t. That's an intrinsic terminator, something that tells the RNA polymerases to stop transcription. But again, I didn't talk about that in your class, so don't worry about that detail for the test. So, again, focusing on the lay of the land here. The promoter, this is what an RNA polymerase is going to be looking for. It's got a -35 sequence, a -10 sequence. It's also got that +1 nucleotide, which is the transcriptional start site. Okay, so this is what RNA polymerases would look for. The operator is the site to which a regulatory protein, I'm going to call it P-LAC-I. P is short for protein. LAC-I is a reference to the LAC-I gene. So the shorthand notation here, I'm not doing it just for today. This is by convention. This means the protein product of the LAC-I gene. The way we talked about it in class, this is the LAC repressor. Okay? So the LAC repressor either binds to the operator or not, depending upon the presence or absence of lactose. And I thought I was clever when I described it to you in lecture. The LAC repressor is being a two-trick pony. One of its tricks is it can bind to the operator, the specific nucleotide sequence of the lactose operant, or it can bind to lactose. But it can't do both tricks at the same time. So if lactose is present, P-LAC-I comes off of the operator, and that allows RNA polymerases to transcribe through and get RNA copies of the 3 structural genes. If lactose levels are low, though, P-LAC-I won't be binding to lactose. Instead, it'll bind to the operator, and it sets up a roadblock that effectively prevents the RNA polymerase from moving off of the promoter. Couple just pretty details that kind of a deeper level of understanding of what's going on here, but still I think pretty cool. I don't know that I even mentioned this in lecture, but the scenario that I've described for you here lets the polymerase be poised for transcription. So it's sitting here raring, ready to go. The instant lactose levels get low, the repressor comes off and you can start transcribing. You don't even have to wait the couple seconds or maybe a minute that it would take for a RNA polymerase to scan along and look for the promoter. It's primed, it's ready to go, poised for transcription. That's cool. It's a testament to how important and what a premium prokaryotes place on this idea of being able to respond quickly to their stimuli. So that's one thing. The other thing, and I did talk about this at some length, is that that promoter is not a very good promoter. And in fact, you'll hear molecular biologists, after they've been talking about this for a while, they use a technical term. They say that promoter sucks. It's not a very good match to what it is that an RNA polymerase is looking for in terms of those 13 or so nucleotide sequence that it likes to see. It's such a bad promoter, in fact, that RNA polymerases are very unlikely to hang out or be attracted by it unless another protein is present. And that other protein does like this promoter. That's cyclic AMP receptor protein, CRP. When cyclic AMP receptor protein is there, then RNA polymerase can look at that promoter, or is interested in that promoter. Cyclic AMP receptor protein provides a link to glucose levels in the cell. When glucose levels are high, there isn't much cyclic AMP receptor protein binding to that promoter. When glucose levels are low, that's when cyclic AMP levels increase, and thereby cyclic AMP receptor protein finds it and then finds that promoter and initiates transcription. And so that's an example of positive regulation. When cyclic AMP receptor protein comes to that party, it's helping RNA polymerase transcribe. It's an upper positive regulation. The LAC repressor protein, P-LAC-I, is sort of the classic example of negative regulation. When it's binding to DNA, it's a downer. It reduces the levels of expression of the lactose operant. It reduces the levels of activity of RNA polymerase. Okay? Is that good enough? All right, I'd be happy to tell you more. I saw her first, but do you have questions about what I've just talked about, or is there new? Great, well, let's deal with that, and then we'll come right to you. The it being the LAC repressor protein. Yeah, P-LAC-I is the thing that would be on the operator. Okay. Yes.
So if you're lactose intolerant, you don't have that?
Okay, so you've kind of pivoted on me here with that question. So in bacteria, in prokaryotes, the regulation of the lactose operon works out like this. So PLAC-I binds to the operator when lactose levels are low. Yes. In humans, when you're talking about lactose intolerance, what's actually going on for lactose intolerance is that a human who's lactose intolerant doesn't / cannot metabolize lactose. And so it stays in their intestines, and bacteria like E. coli use it as an energy source, and they produce carbon dioxide gas, there's bloating, there's smell, diarrhea, all the rest of the stuff that comes when you give a person who's lactose intolerant a dairy product, which has a lot of lactose in it. All right? For people who are not lactose intolerant, what that means, for people who are lactose tolerant, I suppose, what that means is that we can metabolize. I am not lactose intolerant. I can metabolize lactose apparently just fine. And that's not the normal state of affairs for mammals. Like, it's a rare dog that is lactose tolerant after it gets weaned, after it's no longer a puppy. That gene for the equivalent to beta-galactosidase shuts down because dogs don't ever expect to see lactose again after they've weaned off of their mother. Right? So the normal state of affair for a mammal, I'm a mammal, you're a mammal, dogs are mammals, cats are mammals, the list goes on, is after you've reached a certain stage in your life, you stop expressing those genes. And so a repressor comes along and shuts that off. People who are lactose tolerant actually have a mutation that, and it's a double mutation. They have two copies of the mutation, one to their mother's version of the gene, one to their father's version of the gene, that prevents them from turning off the lactose operon later in life, the way that we had done before the advent of dairy farming, about 15,000 years ago. All right? Probably a lot more to an answer than you wanted, but if you're lactose intolerant, if you are lactose intolerant, what that means is that you are stopping the expression of the lactose operon from your genome. All right? But that results in a lot of lactose in your gut if you've had dairy products, and the bacteria in your gut are not lactose intolerant, and their repressor comes off the lac operon and allows it to be expressed. Kind of fun. All right.
I was wondering if you could show us how to do number 60 on the
I think I need to see it. This is the one from last year? Great. So she's asking about question 60 from last year's final exam. And it's a multiple choice question. So I'm going to kind of cop out on you here, and I'll tell you that what we're looking at, and the math works out pretty easily. So we've got an F2 generation where there are 50 green smooth plants, 30 yellow wrinkled plants, 10 green wrinkled plants, and 10 yellow smooth plants. The total number of progeny in the F2 generation is 50 plus 30 plus 10 plus 10. Somebody was looking up for you, and it adds up to 100. So the denominator is going to be 100. The numerator for the recombination frequency now needs to be the number of recombinant individuals. And so there are two kinds of recombinant individuals in this question, the 10 green wrinkled plants and the 10 yellow smooth plants. 10 plus 10 is 20. That's my numerator. Those are the number of recombinant individuals. They don't look like either the mother, they don't look like either the grandmother or the grandfather that gave rise to this lineage. And so 10 plus 10 divided by 100 is 20. Yeah. I'm coming. Yeah. I sent somebody an email yesterday afternoon, and then I saw them like an hour later, and they said, gee, you know, I got your email, but I've been busy and I haven't had a chance to read it. And I said to them, yeah, I know, a lot of words, right? And I think the same thing could be said about most of these exam questions. Like, really, you have to read the whole thing? It turns out it's going to come in handy for you tomorrow. So her question's about number 1, also from last year's final exam. And we're talking here about some work that Thomas Hunt Morgan did. He found out that cinnabar and black body recombine with a frequency of 9%. Cinnabar was also 9.5 map units from the gene for vestigial wings. And the question is, what can you conclude about the genes for black body color and vestigial wings? So I'll tell you, just kind of facetiously almost, these are the things that come to mind. What could I conclude about those genes? At a first pass, I can conclude these genes are on the same chromosome. I don't think that's a particularly sophisticated conclusion, but that may be all that I'm gunning for on this particular question. We'll find out if that's among our choices. But I know they're on the same chromosome because cinnabar and black body are clearly on the same chromosome. They have a recombination frequency that's less than 50%. And cinnabar and vestigial wings are also clearly on the same chromosome. So vestigial wings, cinnabar, and black body color all have to be on that same chromosome. It's a tautology almost. But I'm afraid that that's not going to be one of my choices. Let's see if it's one of my choices. Because if it's not one of my choices, I have to think a little bit deeper, not that much deeper. But the first choice is the genes are on different chromosomes. That's wrong. I'm moving on. They recombine with a frequency of either 0.5% or 18.5%. And I'm talking specifically about cinnabar and vestigial wings. And I can tell you that's going to be the right answer. Right? They're on the same chromosome, and I know that's the right answer, and I can talk with you about it, but let's explore why these others are bad. See, the two genes have an epistatic or pleiotropic relationship. Those are words we kicked out into class, epistatic and pleiotropic, but I don't see how it's at all relevant to what's going on here. Pleiotropic, a single gene's affecting multiple characteristics. There's no whiff of that going on here. Epistatic, one gene's affecting the expression of another. Gee, if that's the case, that didn't come at all from what was provided as part of the question. D is body color and wing size must be determined by a single gene. Well, that sounds kind of crazy because I'm hearing that there are separate genes for each of these three things. So I'm not too keen on D. And then E, the vestigial wing gene must be located closer to a centromere. Well, that's just out of nowhere. You know, that centromere is, okay, I know what a centromere is, but I have no basis for telling which of these three genes might be close to a centromere. That's just a sucker, plausible answer. That's for somebody who has absolutely no idea what the right choice is. They might gravitate to that one just by guessing. But here, B is the right answer. They recombine with a frequency of either 0.5% or 18.5%. And so we got three genes. I got black body color and cinnabar eyes, and I am told they are 9% recombination frequency. And I have cinnabar is 9.5 from vestigial wings. But I don't know if it's 9.5 in that direction or 9.5 in this direction from the information that's been provided here. And so it could be it's going to be one or the other of those. That's what it comes down to. If it's 9.5 to the left, then vestigial and black body color should be about a half a recombination unit apart. If it's 9.5 to the right, then it's 9 plus 9.5 or 18.5, and hence B. Sure. Also last year's test? Could you tell if I showed it to you? Nah, don't bother. It's okay. It's probably the same question anyway. Okay, so this is question 29, either off of last year's test or some other. Okay? And it's about the HFR mapping of genes on E. coli's chromosomes. So what you get is a set of 5 HFR gene transfer orders. And I'm just going to start with the first one, just because it's at the top of the list. I have a suspicion it's probably not the best one to start with, but you could start anywhere and we'll make do with what we do. So I know from one HFR strain that E. coli's got some genes on its chromosome that are in this order. And I am just copying this straight off of the exam itself. And I think, boy, that's nice. I got a good chunk of genes there. Those come from an HFR string that's called J. Okay, good enough. But I want to know the rest of the sequence of genes on E. coli's chromosome. And so what I want to do is I'm just going to look to find one other HFR strain that gives me some information about what's around the lactose operon. I know that after the lac operon is pro and ton and AZI and LU, but what comes before? And so, do I see lac anywhere else? And sure enough, I do. AB221, the last one on my list of 5, tells me that on one side of the lac operon is pro, on the other side is ADE, on the other side of that is GAL, other side of that TRP, other side of that HIS. And so if you're wondering what's going on here, what must have happened is that when the JF plasmid inserted into the JHFR chromosome, it was pointing one direction, and when the AB221 F plasmid inserted into its bacterial chromosome, it's pointing in the other direction. So one's telling me what's on one side of the lac operon, the other one's telling me on the other side. And again, this comes from AB221. I also get that from AB221. So now I want to know what's on the other side of HIS. I know on one side is TRIP, but what's on the other side of HIS? I'm just looking through the list here. There must be another HIS here somewhere. Sure enough, AB219 tells me that on one side of HIS is TRIP and GAL. It's good to confirm that. But on the other side is STR, MAL, XYL, and MTL. And so this comes from AB219. All right, so what's on the other side of MTL? And the other side of MTL. The other side of MTL, strain K gives me some information. On one side is ILE, on the other side is XYL. Here's XYL, MAL, STR. So I need to know that this is ILE. Then I got MET. And I've got THI. So this comes from strain K. And what else is going on with THI? On one side is MET and ILE. On the other side is THR. And on the other side is LEU and AZI. So I've come around the circle now, and 9 gives me information from ILE all the way through there. And so the hard work is done. I think at this point we've mapped the whole circle of E. coli's genome. And the trick now is just of the 5 choices that you've been given, which one of those is consistent with one of the orders of genes that you see on that circle. 4 of them won't be, 1 of them will be. Kind of a puzzle. Yeah. What about Tata boxes? Uh-huh. Yeah. Sure. So, did you say question 36 off of 2013's exam? So that question is diving in a little bit deeper on what's going on with this promoter. I've already mentioned it this morning that the promoter has some sequences at -35 and -10 and that +1 nucleotide. That's what the RNA polymerase is looking for to say, this is where the starts of some genes are. I should sit down here and start transcription. The question, 36 from 2013, is just asking how many nucleotides are we talking about? How big is that -35 and how big is that -10 sequence? In lecture I mentioned they're 6 long each. They're each 6 long. I tried to make it relevant by reminding you that that's a total of about 13 nucleotides. That's about how many nucleotides you would need to look at to be able to be confident that you didn't stumble upon it by chance. If a promoter was simply the nucleotide A, if that's all it took to get the attention of an RNA polymerase, literally every 4th nucleotide you'd start to initiate transcription because there are 4 nucleotides, so it's got to be bigger than 1. There would be, if E. coli's genome is 3 million nucleotides long, okay, let's just make the math easy. If it's 4 million nucleotides long and every 4th nucleotide is a promoter, you're going to have 1 million promoters. That can't work. There aren't 1 million genes. All right? If it's 2 nucleotides long, it's going to be stopping every 16th nucleotide pair. Still too small. It's only when you get up to around 12 or 13 that you're at a level where you can be confident that that's not a sequence that just happened by chance. That really is something special. It's one of a kind. That's got to be the promoter for a gene. Right? And so again, it just boils down to each of those are 6 nucleotides long. It's also consistent with something I mentioned in the lectures about how it is that DNA binding proteins go about recognizing sequences. They lay an alpha helix across one of the major grooves. And if you look at a major groove and you count the number of nucleotides, you can see it's 5 or 6. So that's mechanically also what's going on. Or you could just memorize 6, and you're all set. Okay.
This one goes to, it's asking, a hen is heterozygous for a sex-linked recessive lethal gene. What ratio of males to females will be produced when she is crossed with a normal rooster?
Okay, so there's some people who are joining us today virtually. So we're doing this online as well. So the hen, we got a hen who's heterozygous for a sex-linked recessive lethal gene. Okay, that's the premise for this question. And the question then asks, what's the ratio of male to female or female to male? I'll figure out male to female, and then if it should be female to male, it's easy to have you guys figure that out on your own. So we got a hen. She's female, and she's got, they name the sex chromosomes differently in chickens than in birds than they do in humans, but for the sake of simplicity, I'm just going to call her XX. That's the kind of nomenclature we would use for a mammalian sex chromosome. But I'm telling you, she's recessive lethal for one gene. All right, so let's do that. XX, that means she's female. On one of those X chromosomes, she's got something that's bad. Okay? And she has chicks with a male, a rooster, I suppose we call them. Showing off my agricultural chops here. Right? So this rooster is XY in human terms. Okay? So if this rooster packages up a sperm with an X chromosome and it hooks up with an egg with this X chromosome, you're going to get a baby chicken that's got two X chromosomes. That's going to be a female chicken, right? And it should be just fine, no problems with this female chicken. If this rooster packs up a sperm with this X chromosome and it hooks up with this X chromosome, you're going to get something that looks like this. A heterozygous recessive lethal, just like dear old mom, but female. Two X's, one good X, one bad X. But because you got the good, it overwhelms the effect of the bad. That chicken's fine. And female. You package up this Y in a sperm, and you hook it up with this X, and you get yourself an XY. That looks an awful lot like the dad, right? One X, one Y, so it's male. It's a good X, no problems with that X, so he's fine. But if you package up this sperm with this X, you package up this Y in a sperm, and you hook it up with an egg that has this X, what you get is X with the bad recessive lethal gene and a Y. Now, XY means male. However, in this instance, there is no good X to take the place or to override the effect of that X with the recessive lethal. It's recessive, but there's nothing to dominate it and make it not manifest itself. And so this male isn't going to be with us. We're not going to see that guy, literally, because he had a recessive lethal on his X chromosome. And so the ratio here between this heterozygous female and this relatively normal male, as best as we can tell, is you're going to have two females for every one male. And if you want the ratio to be male to female, it's 1 to 2, right? So depending on the question, that's what's up. Good enough? All right. Anything else? Yeah. So question 60 off of the 2013 exam gives you some data from one of Thomas Hunt Morgan's experiments. I picked up eye color, and then there's something else, some other trait that's being considered. And then you get the number of some progenies in an F2 cross. That's what it comes down to. The total number of progeny is all four added together. That's the denominator. Okay. The numerator here is the fraction, it's the number of individuals that are recombinant. And the trick to recombinant is they've got a combination of traits that are different than anything that you saw in the grandparents. And so in this instance, it's those two small numbers are the ones that are new under the sun. Nobody in that family had looked like them before. So I think the numbers were 10 and 12. 10 plus 12 is the number of recombinant individuals. The total number of individuals is the total number of individuals. So you add all four numbers together. The recombination frequency is that simple math. It's the numerator divided by the denominator. That's the recombination frequency. When you express that in terms of percent, that translates it to map units. One map unit corresponds to 1% recombination frequency. Okay? For questions like that, it's my inclination to make the math easy or unnecessary. All right, so that one, it sounds to me like you may actually need a calculator, but I hope the choices were such that, no, these are just obviously incorrect, and this one's in the ballpark.
So what are like the definitions between positive regulation control and then negative?
So the question is, what's up with positive and negative regulation of gene expression? You asked for a definition. I'm going to tell you straight up the definition, no long-winded story. Positive regulators are proteins that, when they bind, help RNA polymerase cause a gene to be expressed. Negative regulators are proteins that, when they bind, make it hard or harder for a gene to be expressed. The LAC repressor and the TRP repressor are negative regulators. Cyclic AMP receptor protein is a positive regulator. Sure. On what? Okay. Okay. So she's asking about complementary strands. And here's the things that I think are kind of relevant or interesting about complementary strands. So keep track of the orientation of DNA molecules. For the rest of your life, if you get confronted with a DNA molecule, notice where its beginning and where its end is. The beginning is the 5' end. And I'm just going to write out some random letters, G, A, T, C. That gives me a good mix. And then G, G, G. How about that? So the 5' end is there. The 3' end is there. If this was a double-stranded DNA molecule, the two strands are anti-parallel. They're pointing in opposite directions. So I can fill that in right away. And the nucleotides are complementary to each other. So for a G, there's a C. For an A, there's a T. T, there's an A, C, G, and then C, C, C. All right? So we've got a double-stranded DNA molecule. Now, if you're wondering, if you feel like there's a question that's confusing you, I'll bet what happened is this, is that you lost track of the 5' and 3' bit. And so if I were to ask you which of these is complementary to this, you know, one of the answers I'll probably, if I want to trip you up, and that's kind of my job in these tests, is to find ways to trick you, right? One of the things I might do is how about You know, I can, how about, well, just this, right? This is not complementary to this in a double-stranded DNA molecule kind of way, right? And what I could also do is I could say 5' to 3' CCC GATC 3'. I could leave this off of the list of choices and give you this. And this is, in fact, complementary to this. Because notice what I've done is I just took this and I wrote it up here, but now 5' to 3' is 5' to 3' that way. So this is actually complementary to that. It's, again, the orientations of that 5' and 3' that I think are probably the biggest trick to that question. So there's two things I'm asking about on that question. There are two concepts that I'm pinging on. One of them is, do you know that for every G there's a C? And that's, you are all over that. And then I'm also checking, do you know the importance of the orientation? And that's another sort of level of wrestling with this question. And if you're going through the questions quickly, you might miss that and bite on an incorrect choice. $45? $47. All right. It's from 2016. Great. All right. So number 47 from last year's test is what this question's about. Talking with you about the restriction enzyme HEPA2. Give you its recognition site. It's 4 nucleotides long. I tell you the human genome is 3 billion nucleotides long. Into how many pieces would you cut the human genome if you turned HEPA2 loose on it? So, the way you figure this out, thankfully I give you some of the math here. B is the right choice, but the way you figure it out is you've got a genome that's 3 billion nucleotides long. And if I asked you how often would you encounter an A, you would say, oh, every 4th nucleotide on average would be an A. And if I asked you, well, so what fraction of the nucleotides in the human genome are A, you would take 3 billion and divide it by 4. But I'm not asking you how often you'd encounter an A. I'm asking you how often you would encounter CCGG, a particular set of 4 nucleotides. Those 4 nucleotides will only occur every 256th set of 4 nucleotides that you look at. Right? 1 nucleotide every 4th, 2 nucleotides every 16th, 3 nucleotides every 64th, 4 nucleotides every 256th set of 4. So 3 billion divided by 256 is what I give you as part of the answers here. And I tell you, I've done the math with a calculator apparently, and I tell you roughly 11.7 million times you would find that in a genome 3 billion nucleotides long. What's that? Every 256 sets of 4. You can make 256 different combinations for nucleotides. Yeah, it's binary kind of numbers. You know, there's a clock on the wall behind me here, and I'm frankly shocked that we've been at this for about 55 minutes. I mean, I feel like I've been talking for about 15 minutes, so I'm like in some sort of time warp here. Maybe that means I'm having fun. It probably means I'm giving you much longer answers than you care to hear, but.
Patrick, I really quick one from online.
Sure.
Is
Yeah.
And are the questions taken from past exams?
So for those of you who didn't hear, how similar is tomorrow's test to the ones that you'll see on the pilot page for the course. I found that I spent more time working on this year's test than I typically do. And so that translates very directly into this exam is going to look more different than the other pairs of old tests that you'd looked at. So if you looked at 2014 and 2015, you'd look at it and say, oh man, like 80% identical. This is going to be more different because I had more time to work on it, or better, I took more time to work on it. But I did start with the 2015 test. I literally copied it as a Word document and I pasted it into a new Word document. So that's the starting point, and then I went through question by question. I cut 2 out because this one's only got 58 questions as opposed to the 60 that I've had in previous final exams. It's that pre-quiz that you guys took before the semester really started that accounts for that difference. And then I tried, if there were things that I thought I spent more time talking about this year than other years, I tried to have the emphasis of the tests be more on those sorts of things. And when push came to shove and I had to finish the whole thing up and I was still not happy with some questions, then I took questions out from the 2015 test and I tried to find suitable substitutes or plausibly good ones from old tests. And so for some of these, like you guys have been asking me some questions from old tests, I look at them and think, oh, I liked that question. I should use that question again sometime. That's where a lot of the new questions came from. So, short answer, I think the previous exams will be a really good guide for you about what you'll be seeing tomorrow. I think it's a fair test. There's, if you look at the old tests, you'll be well prepared. I'm hoping that when you guys are looking at the old tests, I know how this goes, right? When you're looking at the old tests, your first pass through the old test is like, what's on this test, right? And what was the right answer the last time it was on this test? Because that conceivably will save me some time when I see it tomorrow. Got it. But if you scratch a little deeper, I hope what you'll do is you'll start looking at multiple tests and you'll say, hey, this question comes up every year. I think this question's going to be on tomorrow's test. I better make sure I know what's going on. And is it always the same answer? Obviously, I think that's the way this should be done. And then what's going to start to happen is you start looking at multiple tests. You'll say, hey, this question came up 2 or 3 times, but I thought B was the right answer, and yet here it's D. What's going on? Clearly, and then I know exactly what happens. Clearly, there's a mistake with the answer key, to which I categorically tell you the answer keys that you guys are seeing are correct. Right? There have been literally hundreds of people who have looked for mistakes in those answer keys very aggressively over the years because it's having an effect on their grades. You can imagine. These are very carefully vetted answer keys at this point, now that many people have looked at them. If there are mistakes on those answer keys, I think they've been caught years ago. So, the answer key's right. You'll just have to accept that. And what that means then is look back and see if you can figure out what's different between those 2 questions. Did the answers change? Did the questions change? Did the order of the answers change? Why is it that B was right one time and D was right the other time? And when you start to figure that out, why it is that B was right once and D was right the other time, when you figure that out, there's like a light bulb that comes on, I imagine. There's like a thunderclap, because now you know what I'm asking about in the question, and you can take it to another level. You can say, how could I make C the right choice? Right? What change do I have to make to this question or to the answers to make D incorrect and C correct? Right? That's when you know you got this stuff down. And I think you can just go to bed at that point, put it to rest. You know what's going on. If you're at that level of looking at those old tests, you got nothing to worry about. It's far enough off that I actually started working on making this final exam about 2 weeks ago. And so it's the things I was talking about 2 weeks ago. Stuff like the lac operon. I say, ooh, I want to make sure I talk about this so I can ask this question. And you may have noticed, was it the last lecture? I think it was the last time we met last Thursday. I said, all right, well, I guess I can stop now because these are all the things that I'm going to ask you about on the final exam. There was more stuff in the lecture outline, you might recall, and I said, I don't even remember what that thing was, but I said something and I said, okay, I'm done. We can walk away at this point. The class period was over, but that was a point that I wanted to make sure I made because I knew I had a question about that on the final exam. I think the lac operon would be a good thing to focus on. Beyond that, off the top of my head, I don't have a clear recollection. Your guys' assessment will be as valuable as mine, talking with you right now. I remember starting working on the test when I was talking about the lac operon. Yeah.
This is a more specific question. There's a question on one of the tests that talked about set exons. Could you explain those?
I didn't talk about that at all this year. In fact, I think I talked about that one year and I decided I don't want to talk about that anymore in this class. So I'm not going to ask you anything about cassette exons. No. Yeah.
In terms of the other material, the other exams, should we be basing it off of the previous exams or like the previous question on the exams on the past exam? Or should we be going to look more back to the other exams? It's only 8 questions, so.
So you're talking about the cumulative questions. I remember looking at the first exam you guys saw and the second exam you guys saw, and I remember thinking, oh, I think a lot of people had a hard time with this question. I better put it on the final exam to keep them honest. All right? Yeah, that part I don't remember. And I remember it was for 1 or 2 off of the first test and 1 or 2 off the second test. And then the rest are just like recycled old questions. Yes. Yes, indeed. I did draw some chemical structures for the cumulative part. And so there are 2 questions that have chemical structures that I put on because I remembered that people struggled with those on the first test. So yes. I don't remember those structures. But if you look at the first exam, you've got a hard copy of it, and I think there's a PDF of it on the pilot page that has those structures. That'll tell you what I'm looking for. That's not the exact same question. I had to redraw these things by hand, and I didn't have the PDF for the old exam there. I just had the blank spaces, and yeah, people struggled with that, so I'll ask it again. The exams are already printed. The ship has sailed. But if you look at the structures from the old test, that'll help you. It certainly gives you the sense of what I'm after. Yeah. Mm-hmm. Sure. So if a solution has pH 12, is it pH? I'll do it either way. If it's pH 12, that translates directly to a concentration of hydrogen ions of 1 times 10 to the negative 12th molar. 1 times 10 to the negative 12th moles per liter. That's what the M is, moles per liter, molarity. And it's moles per liter of hydrogen ions. If it's pH 12, I know its pOH will be 2, because pH plus pOH will always equal 14, at least for this class. And you can make solutions that follow different rules, but you need to work to make those solutions. So for typical, normal living systems, pH plus pOH equals 14. So if pH is 12, pOH must be 2, and that translates to a 1 times 10 to the negative 2 molar hydroxide ion concentration. And I think your question was, what about pOH 12? What's the pH? It's just flip those around and you're there. There comes a point in every review session where I think we're at. I'm sensing we're in the home stretch. Nobody has a burning question right now, but there will still be more. I also know that from past experience. And so, you know, in the next 5-10 minutes, tell me what else is on your mind, and let's see what I can help you with. If you're wondering, do I bail now or do I stick around? I think if you decide to stick around, you're sticking around for like 5 or 10 minutes, and then everybody's going to leave. And I'd like to leave at 11:45 to make it to where I have to be, but. Want me to review some structures? Well, here's some fairly basic rules. Hydrogen makes 1 covalent bond. Oxygen makes 2. Nitrogen makes 3. And carbon likes to make 4. All right? So if I show you a structure like that's just wrong. Okay, the problem here is that yeah, the hydrogen's fine, the oxygen's okay, the hydrogen's fine, but that nitrogen's missing something, right? And so, you know, something like this, nothing wrong with that. Right? The carbon's got 4 bonds coming off of it, the oxygen's got 2, the hydrogens all have 1. So I think it could be maybe as simple as that, what's going on with that sort of thing. Along that line, so let's get rid of the one that's not a viable thing. Remember, hydrogens and carbons have very similar electronegativity, so they share electrons pretty equally. Oxygen is electronegative. It pulls electrons toward itself. And so the hydrogen's neutral, the oxygen's electronegative, so you're going to have electrons spending more time here than here. And the same is true for the carbon, so you're going to be pulling some electrons there. You're going to get a little separation of charge. So this is a potential place then for water to interact with it and do some hydrogen bonding. And that could make this a more water-soluble molecule. And in fact, the molecule I happen to have drawn entirely by chance, I assure you. I think we call that ethanol. Right? And ethanol is really quite water soluble. And that's why. It's kind of embarrassing that the random molecule I would draw is a drug. One of my favorite experiences as an undergraduate, I had a favorite instructor in a biochemistry class that I was taking, again as an undergraduate. Loved this guy. I've tried to model my life after him, but I remember at the end of one lecture, he was using an overhead projector. You guys may not have ever even seen an overhead projector, but at the end of, no, actually he was using chalk. It was as primitive as that. At the end of one lecture, he just took a piece of chalk and he was showing how proteins could fold up on themselves to make like a 3-dimensional structure, and he just sort of squiggled something on the board. So see, you know, just a random 3-dimensional thing. And he walks away and he's talking, and everybody in the class, like 50 people, it was a huge class, right? Like 50 people were just, I couldn't help it. Everybody was laughing. And it's like, and after like 2-3 minutes, we're getting real close to the other class. After 2-3 minutes, she says like, okay, so what's going on here? Is my fly open and you guys have just realized it? What's the deal? What's happening here? And somebody said, the protein you drew. It's like, and he says, what, is a naked woman or something? And everybody just breaks out laughing, and he turns around like, oh my gosh, this is a little too X-rated. It was like a naked woman that it just sort of randomly squiggled around. And so, whereas I draw alcohol molecules, my mentor, I suppose, draws naked women when he's left to his own devices to subliminally draw stuff. I think the alcohol is marginally more appropriate. So the Exodus has begun. Anything else? I've been telling you guys over and over again, I don't think you should take more than 45 minutes on tomorrow's test. Maybe an hour would be typical for a student. I don't look at it and think, wow, this is a really hard one. I think it's kind of a run-of-the-mill kind of final exam for the Bio 1120 class that I teach. And I'm going to take this as a sign that we're done. If you have questions, come on up and I'll help you out.